Let's talk about epigenetics, shall we? Epigenetics is the study of modifications to chromosomes that affect gene expression or regulation other than mutations to the genome itself. Today, in this video, we will be discussing a specific type of epigenetic modification, histone modification. But before we get into that, let's take a second to talk about histone structure and function. This is where we zoom the camera into the histone so we can take a closer look. Histones arrange themselves into groups of 8 called octamers. Each octamer is made up of 2 of each type of histone: H2A, H2B, H3, and H4. Now, each histone has a tail made out of amino acids, making a total of 8 on the entire octamer. DNA wraps around the octamer and the tails fold over. The tails are positively charged and the DNA is negatively charged, so they're attracted and they connect, holding the DNA in place. Now, let's talk about the first of 2 major histone modifications, histone acetylation. In histone acetylation, an enzyme called acetyltransferase adds an acetyl group to lysine on one of the tails. Now, what happens? Well, the tail raises away from the DNA. The DNA becomes more loose around the histones, and there's an increase in transcription because more proteins are able to access the DNA. Another enzyme called deacetylase comes in and removes the acetyl group from the tail. It looks a lot like the first enzyme. And make it look different. There. It's a little fancy. Similarly to histone acetylation, histone methylation occurs when a methyl group is added to a histone tail. A few key things are different though. For one, instead of just being added to lysine, methyl groups can also be added to arginine. Secondly, histone methylation doesn't just have to be performed by methyltransferase. It can be performed by any number of proteins. And thirdly, while in the case of acetylation, the tail lifts up, methylation can either lift the tail up or hold it down. So, depending on what happens, you'll either have an increase or a decrease in transcription. This can have a major effect when one of the modifications is made permanent. For example, in mice treated with nicotine, the H3 and H4 tails are acetylated, and this allows for increased transcription of the delta phos B gene, which is the metaphorical on switch for addiction. Heritability of these modifications varies from one chromosomal location to the next. Here are the main ones that I've found. Either the modification is added to the histone during synthesis before they even join up to form the octamer, or the modification is added during DNA replication right there at the replication fork, or the modification is added after DNA replication during the S phase of the cell cycle. And there you have it, everything you need to know about histone modification.